Hi everyone. Thanks for joining CMU BME forum. Today I will be presenting my research on the topic of biomimetic IgA neuron growth modeling with neural morphometric features and CNN-based prediction. So neuron is the fundamental component of the brain, and it's responsible for process signals and control various components of the human body. And they are neurodegenerative diseases that are very difficult to treat because once neurons are damaged or lose connection, they can't really reproduce themselves. So, and there has been some approach indicating that neuron growth factors can help prevent and reduce those kind of problems. So it is crucial for us to model and study the growth process. However, the neural growth process is a multi-stage process as shown here, and each stage involves complex boundary change during the growth with unique growth behaviors. So it is a very difficult process to model using mathematical equations. And existing models in the field, they cannot really model all of those different complex growth stages at the same time. And so there's a need for a simulation tool to do all those different aspects at the same time together. So in our approach, we use phase field method to model neuron growth because phase field method is uniquely situated to solve complex interface change problem because it allows us to avoid explicitly tracking the interface. And for neuron growth, we discretize the domain into zeros and ones, where one represents the cell and zero represents the surrounding environment. And in our approach, we incorporate experimental features extracted from experimental cultural images done by Ashley Liao, another PhD student in our lab. And from the experiment image, we can extract neural morphometric features, and we incorporate those neural morphometric features into our IGAC neuron growth model, so that we can determine the days in vitro, and also to figure out the potential neural tips, as well as adjusting those tips to reflect the neural growth behaviors. So to do those things, we introduce a algorithm called automatic neural tracing, so that we can figure out the total neural length based on tracing three generations of neural tracings based on from geodesic distance. The algorithm is able to take the neural growth field phi and output all those neural tracings automatically. Another algorithm we introduced to achieve the tip detection is called neural tip detection. It takes the neural growth field phi as input, and it's able to automatically detect all the potential tips in the field based on a convolutional box filter. And this allows us to get all those potential tips in the domain. Then to figure out which tip is the ones we want to grow out, we use this mechanism called external cue guided mechanism to mimic the growth behavior of external attractive cue during the neuron growth process. And this mechanism essentially modifies the location of growth cone to attract neuron to grow towards the external cue. And with this model, we are able to simulate the result with and without features. And as shown here, the results with features are actually statistically comparable to experimental results. We apply the model to multiple neuron configurations to show that our model can handle multiple neuron configurations without any issue. And we also build a CNN autoencoder model as a surrogate model to provide us with fast and accurate prediction of the neuron growth process. And so in conclusion, we are developing this neuron growth computational pipeline to provide good realistic results while significantly reducing the computational time. Yeah, thank you.